Assalamu alaikum, dear friends. Today we will be talking about PCR or polymerase chain reaction. It is an important topic from the examination point of view of class 12th, and it comes under the processes of recombinant DNA technology. So let us start. Amplification of gene interest using PCR. So first of all, we will try to define what is polymerase chain reaction. Polymerase chain reaction is best defined as the DNA replication in vitro. As we all know that replication of DNA takes place within the cells at, at S phase of the cell cycle when cell is about to divide. But we can also amplify this DNA outside the cell, outside the natural environment of a DNA, outside a cell in the test tube. Such a technique by which we are able to make several copies of DNA is called as polymerase chain reaction. The technique was first developed by Carey Mullis at CETUS Corporation in California in 1985, which won him a Nobel Prize in Chemistry in 1993, along with Michael Smith. The technique has made it possible to synthesize large quantities of DNA fragment without its cloning. So normally what we do in biotechnology is that if we are to make several copies of a gene of interest, what we do is that we make a recombinant DNA or rDNA molecule, then insert that rDNA molecule in the host cell where it multiplies and forms several copies of it. But in case of polymerase chain reaction, we are able to make several copies of this DNA molecule without even utilizing recombinant DNA technology, without using a cloning vector. Another important aspect, another important benefit of or utility of polymerase chain reaction is that it is ideally suited where the quantity of biological specimen is very low. Say for example, if we obtain a sample or a DNA which is in the form of a single fragment of hair, like hair, or a tiny bullet stain at the crime scene. Even with this small amount of sample or DNA, we are able to amplify this DNA or make several billion copies of DNA out of a single copy of DNA by making use of this very technique, that is polymerase chain reaction. So now let us move on to what is the basic principle, or on which principle does this PCR depend? The basic principle underlying this technique is that when a double-stranded DNA molecule is heated or to a temperature, the two strands will get separated, and each strand will act as a template for synthesis of the new strand. So suppose we know that DNA molecule, it is double stranded, and this double stranded DNA molecule, here if we see this double DNA molecule, one of the strands has an orientation of 5' to 3', and the opposite strand has just the reverse orientation, that is 3' to 5'. So what the basic principle of PCR is that if we are somehow able to separate these two strands of DNA molecule Then each strand, each separated strand, say for example here, suppose we have separated these two strands, this 5' strand to 3' strand, and the another strand, that is 3' strand, 3' strand to 5' strand. We are able to separate these two strands. Then each separated strand will act as template, wherefrom we will be able to synthesize the another strand, so that from one molecule of DNA we are able to form two molecules of DNA. If this process is repeated n number of times, we will be able to obtain, after n cycles, we will be able to obtain 2 to the power n molecules of DNA. Suppose we began with a single molecule of DNA like this, then after one cycle we will be able to obtain two molecules of DNA, number one, and after n number of cycles, the number of DNA molecules which we will obtain will be equal to 2 to the power of n. Now, details how this PCR technique is done, or what are the various techniques which are involved in this polymerase chain reaction. The details of this PCR technique and its mechanisms were described by Ehrlich in 1989 in his book Polymerase Chain Reaction Technology, or PCR technology. The PCR now is fully automated. Nowadays, entire process of polymerase chain reaction has been automated, and today sophisticated machines capable of performing all the steps of PCR are available. Here is one such example of a PCR machine which is used to amplify a given sample, a given molecule of DNA into several billion copies. What are the basic requirements of PCR? The PCR requires the following. There are three important requirements of PCR. Number first is the template, DNA template. Number second is primers, and number third is the enzymes. Now, DNA template. Any source that contains one or more target DNA molecules to be amplified can be taken as a template. So, in other words, we can say that the source DNA or the gene of interest or the fragment of the DNA which we want to amplify, or which we want to make several copies of which, or which we want that it should have several copies, we want to amplify it, we want to make several copies of it, that DNA, each strand of that DNA will act as a template. Number second important requirement of PCR, or polymerase chain reaction, is the primers. Now, primers, as we know, in this replication process in vivo, during DNA replication, DNA polymerase adds the nucleotides only to a primer. Now, in vivo, the primer is actually an RNA molecule, but in case of polymerase chain reaction, the primers to which a DNA polymerase adds nucleotides is a DNA molecule. So here, as you can see, primers, we use a pair of primers, two primers. These are oligonucleotide sequences. Oligonucleotide sequences means that these are DNA molecules formed of small stretches of nucleotides. Say for example, here the number of nucleobase pairs in the primer is 18 to 26. Now actually the optimum or the maximum length of this oligonucleotide or primer, it depends, it should not be too small so that it is inefficient, it is not able to specifically bind to or anneal to the template, neither should it be too long so that it is binding or annealing to the template takes much larger time. So therefore the most suitable size or optimum size of this primer, it is in the range of 18 to 26 base pairs. Now what do these primers do? Primers actually provide the free 3' hydroxyl groups. The hydroxyl groups, the deoxyribose which is present in the nucleotides, it is 3' position, it is free. When 3' position here, so it is carbon number 1, here this is carbon number 2nd, and this is carbon number 3rd. So at this carbon, this hydroxyl group is free and it can form the phosphodiester bonds with the nucleotides that are added by the DNA polymerase. So primers provide free 3' hydroxyl groups for attachment of nucleotide bases by the DNA polymerase. Primers, in another important point that is to be noted is that here two types of primers are required. One primer will attach to one template or one of the two strands of the double stranded DNA. Suppose again here this is the double stranded DNA, one having an orientation 5' to 3' here, and another having an orientation 3' or to 5'. Now when we separate these two strands, the primers that are required are now of two types. One of the primers will bind with or anneal with the DNA template or the DNA strand which has an orientation 5' to 3'. Again, another important point to note is that these primers always bind at the 3' end. So here is one primer and this is another strand here, 3' to 5', right? So here the second primer will bind at 3' state, just like this. So we require two types of primers. One of the primer is here, this type of primer is called as the reverse primer, and another type of the primer is here, this primer is called as forward primer. Now when they will add the nucleotides, when they will add or they will undergo, when DNA polymerase adds nucleotides to the primer, they will extend the DNA chain towards each other. This will move in this direction, this reverse primer, and this will move in this direction. In other words, they move towards each other. Number third requirement of DNA polymerase, of this polymerase chain reaction, is the enzymes. The enzymes that are required in polymerase chain reaction are called as DNA polymerases. Now DNA polymerases, unlike the normal polymerases which we use, which are used during the normal replication in vivo or inside the living cells, they are different from those in the sense that they are heat labile, they are heat resistant, they can operate, they are able to withstand much higher temperatures above 90 degrees centigrade, while as the normal DNA polymerases, they get denatured at such higher temperatures. So the DNA polymerases which we use in polymerase chain reaction, they are heat resistant, they are heat labile, they do not get denatured, they are unable to get denatured at 90 degree at higher temperatures, they can very well function at higher temperatures. So the commonly used DNA polymerases in polymerase chain reaction are: number first, Stark polymerase. The Stark polymerase is isolated from a bacterium obtained from hot springs called as thermophilic Thermus aquaticus. Thermus aquaticus. So it is isolated from a thermophilic bacterium, Thermus aquaticus. Another polymerase chain, another DNA polymerase which is used is PFU polymerase. It is isolated from another bacterial species, Pyrococcus furiosus. Another type of DNA polymerase that can be used is PWO polymerase. This polymerase is obtained from Pyrococcus YC. Another type of polymerase, DNA polymerase, which is used is the TITH polymerase. This TITH polymerase is isolated from Thermus thermophilus. So, and we can use any type of polymerase. These different types of polymerase, there are many more polymerases as well. Apart from these four, these polymerases differ from each other with respect to, say, their fidelity, that is, and the error, etc., or their exonuclease activity. But the most common polymerase that is being used in polymerase chain reaction is Stark polymerase. Now, let us discuss about the working mechanism of PCR. PCR consists of a series of 20 to 40 cycles. In PCR, some 40 cycles of DNA replication take place one after the another. And a single cycle involves three basic steps. Those three basic steps are denaturation, number first, annealing, number second, extension, which is also called as polymerization. Now, how much temperature we use and how much time is spent at each of these steps, that depends upon the melting temperature of primers. It also depends upon the type of DNA polymerase used, as well as upon the deoxynucleotide triphosphate is used. Our first step is the denaturation. Denaturation is also called as melting of target DNA. So the first step is that the DNA molecule, suppose here the DNA molecule, its orientation is one of strand is 5' to 3' and another strand is 3' to 5'. As you can see here, the first step is to heat it. It is heated at about 92 to 96 degrees centigrade, and resulting at this temperature, these two parent DNA strands, they will separate. The parent DNA strand is separate, so that now the DNA becomes single stranded. We now have a DNA strand having orientation 5' to 3', and another DNA strand having orientation 3' to 5'. Please note that here the hydrogen bonds which were holding these two strands together in the double helical DNA or double stranded DNA, they have been broken by the higher temperatures, and this process is what is called as melting of DNA or denaturation of DNA. Number second step is the annealing. So during annealing, what happens is that the temperature is now lowered. So earlier the temperature of the mixture was increased or raised to 92 to 96 degrees centigrade. Now the temperature is lowered so that the primers, the forward primer and the reverse primer, they bind or they anneal to the each separated strand. Please note that these primers have complementary sequences to that of the template strand, to each end of the template strand rather. Say for example, this reverse primer here, it has complementary DNA sequence, or it has complementary sequence to that of the parent DNA strand at the 3' end. Similarly, the forward primer has complementary DNA sequence to that of the parent DNA strand at again it is 3' end. The binding of DNA primers to the parent DNA, separated DNA molecules, separated DNA strands is called as annealing. And again, important point to note is that the optimum temperature for this process is 50 to 70 degrees centigrade, much lower than the temperature required for denaturation of DNA. Here you can see these were the parent DNA molecules. So there's this red line is the one strand and the purple line is the another strand. Number these lines, these short lines which you are seeing here, which you observe here, these are the different primers. And here these are the deoxynucleotide triphosphates. So what we do is that in the reaction mixture in the test tube in the PCR test tube, what we do is that we take all these raw materials and components together in the single test tube. The target DNA molecule, the primers, and number third, the raw materials or the deoxynucleotide triphosphates, and of course the enzymes, that is DNA polymerases. Now when these will be first heated, temperature will be set at 92 to 96 degrees centigrade. This will result in separation of the parent DNA strands like this. So these have separated. Then the temperature will be lowered to 52 to 60 degrees centigrade so that these primers will get annealed to their respective separated DNA strands. Number third step and last final step of polymerase chain reaction is the extension. This step is also called as polymerization. The final step is the extension, wherein the DNA polymerase or the Taq polymerase obtained from Thermus aquaticus, this synthesizes the DNA region between the primers using deoxynucleotide triphosphates. An important point to remember here is that this Taq polymerase, it functions only in presence, it is dependent upon the magnesium dipositive ions for its optimum activity. So when a magnesium dipositive is present in the reaction medium, it is only then that DNA polymerase will synthesize the new strand, and the temperature for extension is 72 degrees centigrade. That is, the reaction mixture which was earlier at the time of annealing kept at 52 to 60 degrees centigrade, it is slightly raised to 72 degrees centigrade, which is the optimum temperature for extension of the DNA, as can be seen here. So here, this is the second step, annealing process. Here, annealing process. This is annealing process, and these are the raw materials. These are the deoxynucleotide triphosphates. These red colored, is white colored, or green and black colored. These are the four types of the deoxyribonucleotide phosphates, which are the building blocks of the DNA, as we know. So when temperature is raised to 72 centigrade, this dark polymerase, this will then add these nucleotides to the primer according to the sequence present, called as per the sequence present on the template strand. So template, it will read template strand. It is DNA dependent DNA polymerase, actually dark polymerase. It will read the sequence of template strand and add the complementary deoxyribonucleotides on the new strand. And after it is completed, we will have two DNA molecules like this. So this is one DNA molecule. This one, this strand is the parent strand. This strand is this new synthesized strand. Likewise, this is a parent strand, and this is the new synthesized strand. And from one DNA molecule, what we have obtained, two DNA molecules. So this completes one cycle. Now, if we as I told you earlier, that in case of polymerase chain reaction involves 40 cycles. It involves 20 to 40 cycles here, 20 to 40 cycles. So one cycle results in production of two DNA molecules. So if we want to obtain several molecules of DNA, what we have to do is that we have to repeat this process. So again, these two DNA molecules are again subjected to heating. They are heated again. The temperature is again raised to 92 to 96 degrees centigrade. This will result in separation of these two strands. So 4 single strands will be formed, and from 4 we will obtain 8 DNA molecules, as can be seen in this graph. So here, this is the temperature which results at which denaturation of DNA takes place, so 92 degrees centigrade. After denaturation, the two parent DNA strands will separate. Then temperature is lowered to 52 degrees centigrade. This is the temperature at which annealing, or the binding of primers to each separated DNA takes place. Then temperature is again raised. So this here temperature is raised and to 72 degrees centigrade, and at this temperature the dark polymerase or DNA dependent DNA polymerase will add the deoxynucleotide triphosphates to the primers, and will result in synthesis of new strand, what is called as extension or polymerization. Then this process is again repeated. Then the two DNA molecules that were obtained in this step, they are again subjected to higher temperature, which will result in their denaturation or separation. Then again the temperature will be lowered. Again annealing will take place. Then again extension. Then again process is repeated. So here again same thing has been explained. The first step, denature the DNA. Double DNA strand is denatured. That means two strands will be separated. This is done at this temperature, 92 to 94 degrees centigrade, and this process it gets completed in around 30 seconds. Number second is annealing. So primers to single-stranded DNA. Primers are again, they are themselves the DNA molecules. Please remember this. In case of the DNA replication in vivo, primers are the RNA molecules, but here primers are the synthetic or the synthesized DNA molecules. So this annealing, it takes place at a lower temperature. So here 55 in average, it can be 52 even. So this process is also lasts for some 30 seconds. So here annealing takes place. Then temperature is slightly raised, not to the level of 94, but slightly it is raised around 72 degrees centigrade. This is where the extension of the primers takes place. It yields new double-stranded DNA molecules like this. Then again this process is repeated. Again it is heated. This cycle will be repeated. For step 1 through 3, it is repeated 20 to 40 times, which will result in the amplification of DNA molecule from one molecule to several billion molecules. So here is exponential amplification. As you can see, from one molecule of DNA, after one cycle, we will obtain 2 molecules of DNA, like here. And then after second cycle, each DNA molecule will again yield 2 DNA molecules. So we have total of 8 DNA molecules. 2 here, 1, 2, 3, 4, and from 2 raised to the power 4, how many strands of DNA? We will have 8 copies of DNA. 1, 2, 3, 4, 5, 6, 7, 8. And in third cycle, number gets doubled. 1, 2, 3, 4, 5, 6, 7, 8, and 8 and 8 is 16 copies. And after fourth cycle, we will have 32. And after 36th cycle, we will have 68 billion copies of DNA. Again, the same thing has been explained here. So after first cycle, DNA copy number will be 2. From 1, we will obtain 2 DNA molecules. From 2, we will obtain 4 after second cycle. Now cycle number 3, we will have 8. Cycle number 30, we will have 1 billion, and so on and so forth. Thank you. So tomorrow in next lecture, we'll be discussing about the uses or applications of the polymerase chain reaction. Thank you for your patient hearing. See you. Allah Hafiz.